I'll go ahead then. Thank you so much for the invitation, and I am very happy to be today presenting the work which I've done as part of my postdoc in India, and also sort of what my lab is currently working on. So I'm at InStem. I'm in the theme of brain development and disease mechanisms, where we focus both on understanding cortical development or the development of the region in the brain, which is the higher order center. And also we also try to understand in neurodevelopmental disorders how things, what are the mechanisms which go wrong in neurodevelopmental disorders. So as my title says, today I'll be talking to you about how the brain is built and what is the role of transcription factors, or how chromatin regulation or epigenetic factors play a role in building the brain. For us to interact with the world, for us to even perceive the world or do anything with the world, we definitely need this mass which is sits right on top of the head, which is the brain. And in the brain, the region or the area which is called as the cerebral cortex or the neocortex is what allows us to take all the higher order decisions, for instance, thinking, learning, memory, consciousness, decision making. All that happens in this region called the cerebral cortex. And this region or this brain has billions of neurons and trillions of glia. So the neurons are the functional unit of the brain, which are the firing cells, which do all the electrical activity and connect. The glia, all the support cells, which provide the supports to all the neurons, and without the glia, the neurons can't function. So it is the neurons and the glia which are part of this entire network of cells in the cerebral cortex. And we have billions and trillions of them, but then all of them actually are produced from scratch from a common pool of cells called the stem cells. And they are actually called the radial glia in the cerebral cortex. And these glia or stem cells actually need to divide to give rise to enough pool of stem cells so that the entire development or the generation or the building of the brain or the cerebral cortex happens. So these needs to divide properly, and that we call as proliferation of these stem cells or the radial glia. Once they divide, then they need to differentiate, and that's called fate specification. And as I said, all the different kinds of neurons and glia are actually made from this common pool of cells, the stem cells or the radial glia. And all the neurons, they are produced from this, and that's called fate specification. So the stem cells undergo symmetric division. They divide, they give rise to two stem cells. But when they need to generate neuron, they undergo asymmetric division. So they divide, one remain as the stem cells and the other becomes a neuron or a glia. And then these give out axons. As you can see here, the neuron, which is multipolar, has actually become somewhat of there is a polarity here, where on one end you have the dendrites, the other end you have the axons. They need to give out axons which need to connect, and that leads to formation of a functional network, which is shown here in the form of connections between the different parts of the brain. And it is this connectivity which gives us behavior with which we can, interact with the world around us. So today in my talk, I'll tell you how, this progenitor gives rise to different kinds of neurons in the period of development. I'll be focusing on one particular class of neurons. So basically, this progenitor has to give rise to all the different kinds of neurons here marked with different colors: orange, blue, brown, and green, but also switch to start producing all the glia in the brain, again, different kinds. But all of this comes from the stem cells across the period in development. And one of the important factors which regulates this progenitor and gives rise to the different kinds of neurons is the chromatin regulation, which happens within the nucleus of this progenitor. Which is actually interaction between transcription factors and chromatin factors or epigenetic factors, which come together and regulate the target gene expression. So with this regulation, you make sure that you make a functional protein, and the amount of the protein or the presence or absence of the protein then determines what kind of neuron you are making, and ultimately decides what kind of a brain are you making, whether it's functional or dysfunctional. This hypothesis of chromatin or epigenetics or the chromatin regulation actually giving rise to different cell states was proposed way back in 1957 by Waddington, and the concepts of these have been refined over time, or we're getting proofs of this in multiple ways in the brain development. So essentially, he simply said that the progenitor basically can run down two different pathways, and depending on what it encounters, whether it's methylation, phosphorylation, acetylation of the histone marks, it will accordingly differentiate into cell state A, B, C, and D, and that happens over a period of time in development. And the chromatin also keeps compacting over time in development. Needless to say, this is very important to study, because not just chromatin regulation is important, but it's often seen that chromatin dysregulation is thought to be at the root of neurodevelopmental disorders. And what I'm putting here is, time and again, we see that there are mutations in chromatin factors, like I've put it all out here, CHAD8 or KDM5C. These are all epigenetic factors. And mistakes in the mutations in these is thought to be at the root cause, whereas synaptic functions are also another class of genes which get dysregulated in neurodevelopmental disorders. So today in my talk, I'll be telling you how I've studied some of the conserved features and molecular mechanisms in mouse, basically telling you how we studied cortical development in health. But how has this now paved the way for me to study this in the human systems, and to look for altered features or mechanisms, and use the human system as an in vitro disease model to understand how cortical development gets affected in disease. So the first part of my talk is, I'll tell you about the mouse brain and what are some of the mechanisms by which you can generate neurons from these stem cells. So as I said, this progenitor needs to proliferate. When it differentiates, it undergoes asymmetric division. And there are two factors which are very, very important in determining what kind of a neuron or a differentiated progeny this will become. One is, of course, time. The other is location. And in my talk today, I'll tell you mostly about time and just briefly give you a hint about the location. So in development, two things are very important, the timing and the location. And I'll tell you how temporally this process of progenitor becoming a neuron, a class of neuron, is being regulated temporarily. So to all the people out in the audience, this is how a developing cortical primordium or a developing mouse brain looks like. So this is a 3D structure here. And what I'm showing you here is a cross section. So if you cut it face-wise, that's called a coronal section. And if you cut it face-wise, this is how a coronal section looks like. You can see very nicely here that there is this tube. It's actually a tube which gets invaginated and creates these hollow, the ventricles or the tube which is filled with cerebrospinal fluid. And adjoining the ventricle is the developing cortical primordium. So if you look at the cross section here, you can see there is this yellow plus red. That's the neuroepithelium. This is where all the development or the making of all the neurons of the cortex happens. It consists of two zones. You can see the yellow one, it's the proliferating cells, whereas the red one is the postmitotic or the neurons. And at this stage, this is embryonic day 12.5. So the mouse gestation is 20 days, is about 19 to 20 days, 18 to 19 days. And day 0 is when the sperm fertilizes with the ovum. And about 12 days from the fertilization, this is how the mouse brain looks like. It is still full of stem cells or the proliferating cells, and there are very less neurons at this stage. But then within the next 8 or, within the next 13 days or about 2 weeks, this primordium, we call it primordium because it's still proliferating, but developing cortical primordium, which is consisting of progenitors and postmitotic neurons, has to become into this highly architectured 6-layer neocortex. And each layer has a different function. You can see there are different neurons here, neurons in purple, red, green, and blue. And each of this actually has a different function. And so basically this primordium, which is initially very similar looking with, say, one class of stem cells or progenitors, has to become the 6 different class of neurons. And how does this become? And that's like a very fascinating question in developmental neurobiology, that how do you build this laminar neocortex from the progenitors? And what happens is actually a very stepwise process of division of these progenitors into making the different kinds of neurons. So embryonically at say day 11.5, there are a lot of these stem cells. These stem cells, they are in a zone called the ventricular zone or the SVZ zone. They divide on the very first day, they give rise to one class of neurons. These are called the layer 5 neurons. That's about embryonic day 11.5 when the layer 5 neurons are being born. And here, so that you can reference it to the future coming slides of my talk, here it is marked by the transcription factor TBR1. So this is layer 6 in the postnatal cortex. Postnatal as in cortex of a pup, which is about 5-day-old. So you can see here that the layer 6 neurons, which are actually born at 11.5, they look like this postnatally. And the very next day at 12.5, these same progenitors, which were producing layer 6 neurons, switch and start producing layer 5 neurons, which then migrate past layer 6, go and sit on top of the layer 6. That's called layer 5. That happens at 12.5. And postnatally, this is how layer 5, it's marked here with FESF2. You can see this is right on top of layer 6. So this is layer 6 here, that's layer 5. And so on and so forth. You keep building the cortex every single day with a new class of neuron being born. So you have layer 6, 5, then you have 4 marked here with ROR beta. And then by 14.5, 15.5, you produce the upper layers, which are layer 2, 3, and marked here with CUPS2. So one of the very important questions is, how does this progenitor know when to switch, how to switch, and how to produce the different class of neurons? So to understand this, we looked at a transcription factor, LHX2, whose expression, and if you recollect, this is how an embryonic day 12.5 looks, and this entire primordium is full of progenitors. So LHX2 is expressed very well in all of these progenitors of the cortex. Soon, within 3 days of development, LHX2 is still expressed in the white asterisk. You can see it's still expressed in the progenitor zone, but you will start seeing that by 15.5, this cortical plate has now neurons, because all the deep layers have already been generated. The layer 6 and the layer 5 neurons have already been generated. But one can see very clearly that the deep layers do not have the expression of LHX2. And postnatally, this is P7, postnatal day 7, the 7-day-old pup, and this is how the cortex looks like. LHX2 comes up in the upper layers, which is here LHX2 positive, but again, it is excluded out from the deep layers, which are the layer 6 and the layer 5. So this led us to the idea that perhaps LHX2 has got some regulation or something to do with the generation of the deep layers. So to understand this better, we took a mouse mutant of LHX2. What I'm showing you here is a control brain, and you may be already familiar with this from my a couple of slides back. So TBR1 here marks layer 6, FESF2 marks here layer 5, but there are 2 additional markers for layer 5 here, ER81 and ID2. And what am I about to show you is a mutant of LHX2, in which LHX2 has been very specifically removed only from the cortex and only at the time point at which the deep layers or the layer 6 and the layer 5 are being born. And as soon as you see this brain, you will immediately figure out that what is happening in this mutant is the layer 6 is reduced, which is TBR1, and the layer 5 is expanded. You can see the expansion or the increase in layer 5, as you can see the expression of FESF2, ER81, and ID2. And not just quantitative, but even qualitatively, we did a number on this, like how many neurons are changed. And immediately we do see that TBR1 is reduced, CTEP2 is increased. You can see here, these are the numbers that at, say, the number of neurons, which is about 20% in the control brain, becomes very close to 27% in the LHX2 mutant brain. Whereas, so when I say an LHX2 mutant, the LHX2 has been removed from these cells. And the TBR1, there is a marked decrease, more than, greater than 40% of the cells are TBR1. But in the LHX2 mutant, it gets reduced down to less than 30%. So clearly, there is a loss of TBR1 neurons I mean, there is an increase in the CTEP2 or the layer 5 neurons, which is happening at the expense of TBR1 neurons. So we wanted to understand this better, that, what is happening at the very molecular level inside the progenitor, which is leading to such a phenotype. Because all we did was remove one transcription factor, and what we resulted is in a cortex which has more layer 5 neurons as opposed to layer 6 neurons. So to understand this, I am going to tell you what are the neuronal subtypes in the neocortical layers and why are they important, or what are their functions, to get a better clarity on what may be happening at the molecular level or at the level of the chromatin. So as I said, layer 6 neurons are TBR1 positive, and these are the kind of neurons which are corticothalamic. So what I mean by corticothalamic is that so thalamus is a region in the brain. Here you can see in the schematic, this is the cortex. Here, the green ones are the layer 6 neurons. You can see that these neurons actually project from the cortex to the thalamus. Now, thalamus is a relay center in the brain. So it is a center which will actually get inputs from the outside world. So any light which hits your retina, right? So let's talk about the visual pathway. So any light which hits the retina, the cells in the retina convert it into electrical signals. Those electrical signals from the retina come to the thalamus. And from the thalamus, they're taken to the cortex. And from the cortex, the corticothalamics are the ones which again send information or decision back to the thalamus to say, suppose a ball comes and hits you, whether you need to catch the ball or you need to duck. That's the decision which is made at the level of cortex. That comes back to the thalamus via these corticothalamics, and that's the layer 6 neurons. Now, the very next layer, layer 5, actually projects to a very different region in the brain. And one of the classes is, this is called subcerebral, which are FESF2, CTEP2 positive, which I've showed you as a marker in my previous slides. One class of these neurons project to the spinal cord, and all your movement or motor functions are happening via these corticospinal motor neurons. Whereas this layer also has another set of neurons called SADB2, which are callosal. And one class of callosal neurons are ones which project from one half of the brain to the other half, because we need to connect the two halves. So you can see here that the red is projecting towards the spinal cord, whereas the blue is actually projecting towards the other half of the brain. So which means each class not just has a different function, but it's projecting towards entirely different regions of the brain. So here, a bit was already known about how the switch can happen from a layer 6 to a layer 5. What happens is when layer 6 neurons are being born, there are two transcription factors, TBR1 and SOX5, which come together and make layer 6 neurons. In doing so, they actually inhibit FESF2, because FESF2 is absolutely important for making layer 5 neurons. So which means that if you want to make layer 6, you need to inhibit layer 5, and that's how they inhibit layer FESF2 and they make layer 6. But when it has to switch, there are two other transcription factors, SOX4 and SOX11, which actually come and bind to FESF2 locus, activate FESF2. Once FESF2 is activated, it goes back and inhibits TBR1. And now the repression caused by TBR1 is away. SOX4, SOX11 activate FESF2. FESF2 then activates CTEP2. And then the switch goes from layer 6 to a layer 5 neuron. And separately, in a subset of neurons, SADB2 gets activated. SADB2 represses CTEP2, and that's how you distinguish between the subcerebral projection neurons, which are FESF2 CTEP2 positive, as opposed to SADB2 callosal neurons in the same layer. So this switch was known. So in this, we were trying to understand what LHX2 is perhaps doing in this switch. So to understand that, since it's a transcription factor, we performed a technique called ChIP-seq, which essentially means that LHX2, being a protein, goes and binds to a spot in the DNA. So if you do a pull down of LHX2, we'll be able to pull down the DNA and ask the question, what is it LHX2 regulating? So we performed a ChIP-seq of LHX2, and what we saw was LHX2 bang on was binding to FESF2 and SOX11, two transcription factors which are very, very important for making layer 5 neurons. And you can see here, this is the binding of LHX2 on the genomic locus of FESF2 and SOX11. And this qPCR here shows that LHX2 only is bound at its binding region, but not at any controlled region where there is no LHX2 binding site. Right. So with this, we immediately ask the question that, okay, so LHX2 is indeed bound to FESF2 and SOX11, but the moment you remove LHX2, what happens to the expression or the levels of both of these transcription factors? Is there any change the moment you remove LHX2? I mean, what is happening? It's getting upregulated, downregulated, or what is happening in the case of absence of LHX2? So to do that, we went back again to the same mutant, but this time, instead of looking at it postnatally, we looked at it within a day of removal of LHX2. So these are brains which are harvested at 12.5 and 13.5. And you can immediately see that in the control, the expression of FSF2 is at low levels at this stage. But in the absence of LHX2, you can see an upregulation of FSF2 in these brains, which is sustained even up till 13.5. And same is true with SOX11 as well, that you see an upregulation of, SOX11 within a single day of LHX2 removal. You can see an upregulation, and this is sustained even until 13.5. So very clearly, what is happening here is LHX2 is bound to FSF2 SOX11. It keeps the two genes repressed, but the moment you remove LHX2, the repression is gone. They get upregulated. The neurons which are actually supposed to be producing less FSF2 SOX11 are producing more FSF2 SOX11. They become layer 5 neurons instead of layer 6, and that's what actually is leading to an increase in the layer 5 postnatally. So with this in mind, we also wanted to understand further, is LHX2 interacting with some chromatin modifiers to do its job? To understand that, we looked at LHX2 interacting partners, and we did mass spec, and what we found was LHX2 is indeed associated with components of the NUD complex, namely LSD1, HDAC2, and RBBP4. This is IP to show that if you do a pull down of LHX2, we see HDAC2 being pulled down, we do see LSD1 being pulled down, we also see RBBP4 being pulled down. And if you do a reverse IP, like if you do a pull down with HDAC2, we see LHX2. We did a pull down with LSD1, we saw LHX2, and we did an RBBP4 pull down, again we saw LHX2, which means LHX2 is indeed interacting with some of the components of the NUD repressor complex. And then we ask the question that, yes, fantastic, LHX2 is interacting with these components, but are these components also present at the same locus as LHX2 on the FSF2 and SOX11 locus? So this is what we wanted to understand, that wherever is LHX2 on the FSF2 SOX11 locus, can we now see LSD1, RBBP4, and HDAC2? So then we performed individual chips with each of these LSD1, RBBP4, and HDAC2 at 2 locations. One was wherever LHX2 is bound, that's called the LHX2 binding region on FSF2 and SOX11, but also at the transcription start site. And the reason is that the chromatin, it's very well known now in the field, the chromatin actually loops from the enhancer to the transcription start site. And I'll come to that towards the end in my cartoon. So to understand this, as I said, we performed chips with LSD1, RBBP4, and HDAC2. As you can see here, LSD1 is actually bound at both transcription start site and LHX2 binding region as well. RBBP4 was only seen with SOX11, whereas HDAC2 was seen at the LHX2 binding regions of FSF2 and SOX11. So indeed, in some combinations, these are also found on the locus of FSF2 and SOX11. So perhaps LHX2 utilizes the function of some of these to actually do its job of repressing FSF2 and SOX11. So to understand that, what we did as a readout was, is the histone marks in the absence of LHX2 changed because, changed, and then that's what is then leading to an upregulation of FSF2 and SOX11. So to do that, we looked at the epigenetic marks. We looked at 2 active marks, the H3K4 trimethyl mark and the H3K9 acetylation mark. These are active marks, which if present on the genome is expressed, and there is expression from these from the genes. So in the absence of LHX2, clearly we saw an upregulation of this H3K4 trimethyl mark, as you can see with the control, from the control sample compared to a control sample. In the LHX2 absence, you can see an upregulation of the H3K4, both at FSF2, SOX11, at the transcription start site and binding region, whereas the H3K9 at the binding region and at the TSS of SOX11, we could see an increase in the H3K9 acetylation mark. So very clearly what is happening here is that LHX2 is able to change the active marks at the locus of FESF2 and SOX11, the marks are less in the presence of LHX2, but in the absence of LHX2, these marks go up, and the FESF2 CTEP2 locus, FESF2 SOX11 locus is upregulated. That then leads to a change in switch in neuronal class from layer 6 to layer 5. So till now, I told you that LHX2 is necessary for this entire process, which means that if you remove LHX2, you get increased number of CTEP2 positive neurons. But is it sufficient? So if you overexpress LHX2, is it sufficient to suppress CTEP2? And turns out, indeed, yes. To do this, we did a technique called in utero electroporation, where we go into the embryonic bray at 12.5, do the perturbation, electroporate the construct, stitch up the mom, let the delivery continue, and harvest it postnatally. And this is how the brain looks like postnatally. You can see EGFP. These are just control electroporation with EGFP. You can see very nice neurons, which are seen from layer 5, layer 6 to layer 5. And these are how the neurons look like. You can see the projections as well. Here is CTEP2 and here is SADP2. And this is how a LHX2 GFP looks like. And what we saw is, what I'm going to summarize it from the bar graph, is at, when we do the electroporation at 12.5, a lot of these neurons are actually CTEP2 positive, rightly so. Close to about 55% of are CTEP2 positive. But the moment we overexpress LHX2, the numbers are drastically reduced to less than 30%. So which means clearly LHX2 is sufficient to suppress CTEP2. Then we ask the question that whatever these neurons, if they're not CTEP2 positive, what are they now? They actually become SADB2 positive neurons. So which means the neuron which was initially becoming a CTEP2 positive neuron, in the presence of LHX2 actually becomes a SADB2 positive neuron, which is another class of neurons in the same layer. So mere LHX2 overexpression is switching it from becoming a subcerebral projection neuron to a SADB2 positive neuron, possibly a callosal neuron in the same layer. So, with this, the summary of this part of my talk is that LHX2 is bound to its enhancer, keeps the active marks set less, it interacts with all of this. The moment you remove LHX2, perhaps these can't now access this. We do see an increase in the active marks, and then we see an upregulation of FESF2 and SOX11. So this was all about the timing, and in the timing, we have now placed LHX2 to play a very important role in control the timing of this switch from layer 6 to layer 5, where we now know that LHX2 actually represses FESF2 SOX11 to regulate this timing. Now, as I said, is there a location angle to this? Turns out there may be. We have a very brief inkling towards, hint towards that. So what is the hint? So in the cortex, different regions of the cortex are devoted for different processing different modalities. So the front of the cortex, so here this towards the left side is the front, here is the OB, the olfactory bulb. So right at the rostral end, you have the frontal or the motor cortex. Right after that, you have the somatosensory cortex, which process all your touch sensation, followed by the auditory and followed by the visual. So visual is at the back, right? So your frontal or the motor cortex has the maximum expansion of layer 5 neurons. Of course, because if it's devoted to movement, the layer which is devoted to movement is the layer 5 subcerebral projection neurons, or the corticospinal motor neurons. It's important to have them at the maximum at the very front end. And if you look at LHX2's gradient, you would actually see LHX2 is very high caudally, or at the back of the cortex. So which means that perhaps LHX2 is part of a mechanism which regulates how, how many numbers of neurons are produced in which region of the brain. And this sort of gives us the hint. Further studies will look into very carefully into how LHX2 interacts with, signaling centers or other transcription factors to actually regulate this process of deciding the region, which region of the brain becomes what. With this, I will switch to the last part of my talk, or this part where I talk about how my lab is now focused in understanding what are the features or altered features or mechanisms in human, and how this is now paving way for us to understand discovery biology of neuropsychiatric syndromes. So this here is a study which was published in Lancet Psychiatry in 2017, which tells us what is the burden of mental disorders in India. We have a whopping 197 million Indians who are mental illness patients, which means 1 in 7 Indian has mental illness. And when we look at the top causes for these mental illness, way up are neurodevelopmental disorders like intellectual disability, schizophrenia, bipolar, and autism spectrum disorders. So which means that, the burden is huge, because the burden is not just on the patient, but also on the socio-economic, strata of the society, because you need a caregiver for many of these patients as well. So which means the burden is huge. You also lose a lot of your significant portion of your life because of these disorders. So all that put together, the burden, just not in terms of numbers, but even qualitatively, the burden is quite huge. And our understanding of such disorders very, very poor, because we don't have the right, we don't have the right sample to look at all of these disorders. For instance, getting access to postmortem samples, or even, developing brain samples is very, very difficult. So, as I said, from these top disorders, we have a lot of them being neurodevelopmental, and neurodevelopmental disorders are, they're not considered as discrete disorders. For instance, here you have in the yellow box the disorders which are childhood NDDs. When I say childhood neurodevelopmental disorders, these are diagnosed very early during your growing up, right from 3 years to 5 years. Whereas there are adult psychotic disorders which are discovered during teenage years, or these are teenage onset psychiatric disorders. But the field now knows that instead of considering, the field is now moving to a hypothesis where instead of considering them as, one discrete disorders, we know that, these are all coming from perturbations as from the neurodevelopmental age, because there are many risk factors or environmental factors which are overlapping between these disorders, particularly between intellectual disability and schizophrenia, which actually leads to overlapping pathological mechanisms, which is leading to disrupted brain development, which then goes on to show as a disorder in either in childhood or in teenage years. So my lab is interested in understanding the one such disorder, which is neuropsychiatric, which is schizophrenia. It's a disorder of aberrant perceptions, thought, and social connectivity. Many family studies shows that it's heritable, that it's neurodevelopmental in origin, and many studies are from Caucasian populations, and very recently there is a study from African population as well. But we don't have any study coming from the Indian population to understand that, what are the factors, or, what are the features of schizophrenia in Indian populations. And what I want to show you here is till all my talk, I was telling you about mouse brain. In fact, the mouse brain is a fantastic model to understand conserved features, but clearly there are many differences in humans. And one notable feature you can see here immediately is the cortex itself is folded, and you can see all the gyri sulci in the cortex, not just in the whole brain, but also in this cross section. Immediately what you can see is that the mouse brain is smooth, whereas the human brain is not smooth. It's got the gyri and the sulci. And in fact, this when you look at a developing human versus mouse brain, no points for anybody to guess and tell me that, there's clearly a difference between the mouse brain and the human brain cut at the same age of development. You can see here, this is a mouse brain. These are the proliferative zones, the ventricular zone and the SVZ. And clearly in the humans, there is this expansion of this proliferative zone, which we call as the outer SVZ, which in fact has been, is hugely implicated in the neocortical expansion, which we see in humans, which is actually it's one of the factors which leads to the folding of the cortex. So what we need is now a model system, which can be very human specific and give us insights into what can be human specific mechanisms, or what can go wrong in human disorders like schizophrenia. So to do that, we have this very valuable system called iPSCs, which are derived from skin or blood cells, where the skin and blood cells are reprogrammed using Yamanaka factors. And from these, we can derive cerebral organoids, which can then be used to study brain development and understand genetics of the disease, and perhaps pave the way for doing drug screening from an individual patient to understand, how a drug, perhaps also go on to go have some personalized medicine. So to do this, my lab actually utilizes the resources from a program called Accelerator Program for Diseases in Brain Using Stem Cells, ADBS, which is a flagship program here at InStem, where 3 institutes have come together, NINHANS, InStem, NCBS, to put together this program, where what we have is a large cohort of clinically dense families. I'll show you one of such families, where there are at least two clinically affected first-degree relatives. Then there are controls also in the family. Their syndromes are all being studied for the disorder. And to study the genetic and cellular basis, we're doing exome sequencing, making iPSCs from these families so that we can study what are the risk variants present in these individuals and how that can cause the disorder. So, as I said, my lab is interested in studying doing human in vitro disease modeling. So the exome sequencing enables us to identify putative risk variants which could contribute to the disease. And then since we have the iPSCs, we'll be actually modeling them in a dish and then try to understand how the clinical assessments can match the cellular and molecular studies. So this is one such family. As you can see here, this is a clinically dense family. You can see several members here in black are affected with the disorder. And there are also unaffected individuals which act as controls. And this family has both schizophrenia and bipolar disorder. And from the exome sequencing, what we got was that there are risk variants in a chromatin modifier, ZD6, which is not there in the control, but present only in the patients. And with this, we have the iPSCs from all of these patients and from the control. The question we are asking is, we will now, from these iPSCs, make cerebral organoids and do a study, which I just showed you in mouse, asking the question that in these organoids or brain-like structures, mini brain-like structures from human, what goes wrong? Which step in development from progenitor proliferation to formation of a neuron, to axon development, to dendrite, to synapse formation? What is going wrong in these patients? Yeah, sorry. Okay. What is going, I'm just towards the very last slide. What is going wrong in these patients? And also ask the question that how ZD6 may affect the chromatin accessibility or the expression of genes. So that will tell us that, how changes in chromatin or gene expression can affect cellular development and lead to perhaps be the causative of, disorders like schizophrenia. So, I think this study will contribute in our understanding the emerging function of the nervous system, that how mutations or altered epigenetic mechanisms can alter neural development and lead to a dysfunctional brain. So this brings me to the end of my talk. And I would like to thank my postdoc mentors and collaborators, Shubha and Sanjeev. All my postdoc funding, the first part of my talk was all from my postdoc, but these are all my current lab members and all the current lab funding, and I'm happy to take questions.